This is the second of two videos covering the A-level biology topic of monogenic inheritance and monohybrid crosses. In part 1 we learned some new vocabulary and recapped how to draw a simple Punnett square. And in this video we're going to look at how this can be complicated by multiple alleles, codominance and sex-linked traits. A trait is monogenically inherited if it's controlled for by a single gene. If we carry out a monohybrid cross, then we're investigating the inheritance of a single characteristic coded for by a single gene, and we're assuming that the parents are the same in all other aspects. When you first learned about inheritance in GCSE biology, we assumed that every gene had two alleles, and that one allele was dominant and one allele was recessive. Now this is true for lots of genes. The dominant allele is the allele that is always expressed, even in a heterozygote, whereas a recessive allele is only expressed when no other alleles are present, so we're only going to see it expressed in the homozygote. And we can differentiate between these two alleles by using an uppercase letter for the dominant allele and a lowercase letter for the recessive allele. Now that is true for lots and lots of genes, but there are some genes that have more than two alleles, and even amongst those genes that do only have two alleles, it's sometimes the case that neither allele is dominant over the other allele. Let's look first at these genes that have multiple different alleles. Now, the system that you've been using so far, with the uppercase letter and the lowercase letter, only has two options. So that's clearly not going to work for us if we're trying to differentiate between three different alleles. So what we do instead is we pick one letter to represent the gene of interest, and then we use a second letter to represent the allele. But if we drew them as the same size, it would look like we were talking about two different genes, which we're not. So what we do is we place the letter for the allele in the superscript position, so where you would write a squared sign. So for instance, we could have, if we had a flower where there were three different alleles for different colours of that flower, all for the same gene, we could have CY for the yellow allele, CB for the blue allele, and CR for the red allele. Now, as we've said, in the versions we've met so far of different inheritance, you've just had dominant and recessive. So we could say that blue is our dominant allele, and we could say that yellow was our recessive allele, but what are we going to say about red? Well, this is what we call incomplete dominance. So we could say that red was incompletely dominant in that it's dominant over yellow, but not over blue. And so we can put these into a little list and say, well, blue is dominant over red, and then red is dominant over yellow. And this is what we call a dominance hierarchy. Once you know that genes with multiple alleles exist, there isn't anything really more complicated about their Punnett squares. It's just making sure that you're familiar with that nomenclature of using one letter for the gene and one letter in the superscript position for the alleles. So here we have a question about those phenotypes that we just mentioned. We're going to have a blue flower and a red flower, and we're told that they're both carrying the yellow allele, so they're both heterozygous. So we're going to go through our normal 6-step process in order to work out what the phenotypic ratio is. So firstly, we write down the phenotype of the parents, which here is blue and red. Then we write down the genotype. We know that they're both carrying the yellow allele as well. So my blue parent is CBCY and my red parent is CRCY. Then I'm going to write down the genotypes of the gametes. So we have CB or CY coming from the blue parent and CR or CY coming from the red parent. And as usual, I ring those genotypes to show that they are the gametes. Then I'm ready to do my Punnett square. So I insert the gametes around the outside, and then I populate the inner cells there. Then, even though I haven't been asked to, I'm going to write down the ratio of genotypes, which here is 1 to 1 to 1 to 1, because all four offspring are going to have a different genotype. And then my phenotypic ratio, I've already said that blue is completely dominant over red and yellow, and red is incompletely dominant just over yellow. So I'm going to have 2 blue flowers to 1 red flower to 1 yellow flower. Another classic example of codominance is blood type. So red blood cells may or may not have antigens on the outside of them, and there are 4 possible phenotypes that you can have. You can have no antigens at all, in which case you're blood group O. You can have one type of antigen that we call blood group A. You can have a different type which is called blood group B. Or some lucky people like me have both of those types of antigens on their red blood cells, and therefore they are blood type AB. Now the A and the B alleles are codominant to one another, and they are both dominant over the O allele. Now another word for antigen is isoagglutinogen. So the designation for blood type is a capital I, and then within that we have the alleles IA, IB and IO. So a person who is blood type O doesn't have any antigens, and therefore we know that they must be homozygous IO. If a person is blood group A, then they could be homozygous and be IA, but we also know that A is dominant over O, so it's possible for them to be IO. In a similar way, a person who is blood group B could be IB, or they could be IO. And then finally, a person who is blood group AB must have one of each the A and the B alleles. A question might ask you to show how parents who are blood type A and blood type B could have offspring with all 4 possible blood types. So we'll go through our normal process. We start out with the phenotype of the parents. So that's going to be blood group A and blood group B. Now, in terms of possible genotypes, we've said that someone who's A could be IA or IAIO, and likewise someone who's blood group B could be homozygous or they could be heterozygous. However, the question has told us that these people have to be able to produce offspring with all 4 possible blood groups. So that tells me that I'm going to have one offspring who is blood group O, and that person is going to have to inherit the IO allele from both parents. So actually we can cross out the homozygous genotypes because we know that those can't be used in this instance. Next we can write down our gametes. So each parent is going to produce 2 gametes which are different to one another. Then I can do my Punnett square cross. So I have my gametes inserted in the outside bits, and I've put those rings around them to show that they're gametes. So I'm going to have one individual who has a genotype of IAIB, and then one IBIO, one IAIO, and one IOIO. I can annotate my diagram to show what the blood groups of all of these individuals will be. Here's another example of a codominant system so that you can pause the video and have a go at doing a Punnett square for yourself. Let's see how you got on. So your first step was to write down the phenotypes of the parents, which in this instance is given to us in the question. They're both roan. Next we need their genotypes, which is slightly trickier. They've told us that there are only 3 phenotypes, and they've told us that red and white are codominant. So this implies that the third phenotype must be the phenotype of the heterozygote. So both of my roan parents are going to have the 2 alleles, the white allele and the red allele. The gametes are nice and straightforward. We just split the genotype of the parents and draw rings around it to show that these are the gametes. Now I'm ready for my Punnett square. Once I have those genotypes written in, I can look at the individual genotypes and assign them phenotypes. So the individual which has 2 white alleles is going to have a white phenotype. The individual that has 2 red alleles will have the red phenotype, and then the 2 heterozygotes are going to be roan. And so that gives me a 1:2 to 1 ratio. Before we look at sex-linked traits, let's briefly recap how sex is determined in humans. So in humans we have a system where if you have the 2 X chromosomes, your sex is female, and if you have an X and a Y chromosome, your sex is male. You could be asked to show why on average 50% of offspring are female. So our parents' phenotype is that one is male and one is female. Their genotypes are XY for the male and XX for the female. Therefore our gametes are an X and a Y and 2 X's. And therefore we can draw a Punnett square where we do this cross, and we can see that 50% of the offspring are XX and therefore female, and 50% are XY and therefore male. Sex-linked traits or sex-linked characteristics are ones where the probability that you inherit that trait from your parents is dependent on the sex that you are and the sex chromosomes that you have. Common examples include colourblindness and haemophilia. The reason why your sex is going to influence how likely you are to inherit that disorder is that the gene for it is coded for by the sex chromosome. And this tends to be the X chromosomes just because there are more genes on the X chromosomes, but it can also occasionally be on the Y chromosomes. If we consider a certain disorder which is coded for by a gene that's present on the X chromosomes, if a person has XX sex chromosomes and they happen to inherit one faulty allele for that gene, they still have another X chromosome, and that X chromosome presumably has a working copy of the allele. And so it's possible for the faulty allele to be masked, because maybe the enzyme that it's coding for, we can make enough of that enzyme from the working copy of the allele. If we contrast that with someone who has XY chromosomes, the Y chromosome is much, much smaller and it has far, far fewer genes on it. And so it's very unlikely that there's going to be a working copy of the allele. So if that person with the XY karyotype inherits one faulty copy of the allele, there's nothing to balance it out and hide it. Effectively, the male person is acting as if they're haploid. We call this being hemizygous. In order for us to do genetic crosses and draw Punnett squares, we need a nomenclature system that allows us to show that these particular characteristics are sex-linked. So we carry on using our normal XY system in order to show what sex chromosomes a person has, and we then use the superscript system to show what alleles they have for this particular characteristic. If there are two alleles and one is dominant over the other, then we still use the uppercase-lowercase system to show which is the dominant allele. Let's take haemophilia as an example. Haemophilia is coded for by a gene that's on the X chromosome. So if a person is a healthy female with two working alleles for that particular gene, we would show her with XX to show that she's female and carrying XX sex chromosomes, and then we would have the two superscript capital H's to show that she has two of the healthy allele. Then if she were a carrier and she had one copy of the allele for haemophilia, then we would show this with a lowercase h. If we have a healthy male, then we can still see the healthy allele on the X chromosome, but we don't put an allele on the Y chromosome, because haemophilia is coded for by a gene that's found on the X chromosome, not the Y chromosome. And then if we had a sufferer who had haemophilia and was male, then we would expect to see X with a little h to show that he has that haemophilia allele, and then the Y to show that he has his Y chromosome. So let's now put this information together with our skills drawing Punnett squares in order to show how a carrier female can have sons who have haemophilia. So our first step is to write down what the phenotype of the parents is. The mother is a carrier and presumably the father is healthy. The genotype of the mother is going to be XX, and she's going to have both a healthy allele and also an allele that indicates that she's carrying haemophilia. Then for the father, we're going to have X with a capital H to show that this is a healthy allele, and then for the Y chromosome, we don't have a letter indicating whether there's haemophilia or not, because that particular gene is X-linked, not Y-linked. Then we can write down the gametes of the parents, and we ring those to show that they're gametes, and then we can do our Punnett square cross. So as usual, we're going to populate each cell in the Punnett square by looking at the gametes. When we're writing out a sex-linked genotype, we always want to put the X first. So in the same way that you would always say that the male chromosomes were XY, you wouldn't say YX. Here as well, we're going to write them the right way around. And in an instance where we have a heterozygote, we're always going to put the dominant one first, just like we would in writing a conventional genotype. So now we can identify our phenotypes. In the top left here, I have a healthy female. In the top right, we have a healthy male. In the bottom left, we have another carrier female like her mother. And in the bottom right, we would have a male who suffers from haemophilia. Thank you very much for watching, and I hope you're now feeling slightly more confident about drawing Punnett squares to illustrate monogenic inheritance. Next, we're going to move on to dihybrid inheritance and epistasis. If you did find this useful, then don't forget to like and subscribe for more A-level biology videos coming soon.